Okay, I think we can get started. My name is Changyang Hu. I'm assistant professor in the Department of Cell and Developmental Biology at University of Michigan, and welcome to the Single Cell Spatial Analysis Seminar. Today we have our wonderful speaker, Dr. Longzhi Tan. He's an assistant professor of neurobiology at Stanford University. Dr. Tan is originally from Wuhan, China, and he received his bachelor's degree in physics with a minor in biology from MIT in 2012. Studying microbial and human evolution with Jeff Gore and Pardis Sabeti. He earned his Ph.D. in systems biology from Harvard in 2018, developing high-precision methods for single-cell genomics with Sunny Xie. He uncovered the 3D structure of the human genome in a single cell, revealed unique chromosome organization in the mouse eye and nose, and measured the true mutation spectrum of single neurons in the normal human brain. Dr. Tan also attended the neurobiology course at MBL in 2014 and worked with Ibrahim Sise at MIT in 2019. As a postdoc in Carl Deitherow's lab at Stanford Bioengineering, and also co-mentored by Dr. Howard Chang, Dr. Tan discovered major 3D genome transformation in the mouse brain after birth. Dr. Tan started his own lab at Stanford Neurobiology in December 2022 and discovered evolutionarily conserved 3D genome restructuring over the lifespans of both human and mice. Dr. Tan's awards include PSF Mind Award, Sanofi Eye Award, HHMI FHS Finalist, Baxter Award, BWF Cassie Award, ISFS, Berry and Science Life Lab Grand Prize, HHMI ISRF, and IPHO Gold Medal. Outside the lab, he enjoys designing holiday cards, t-shirts, music videos, and is a scientific illustrator. So please join me to welcome Dr. Tan.
Thank you so much, Changyang, for the very kind introduction. And thank you, Single Cell Spatial Analysis, for inviting me to speak. I'm very excited to give this presentation as Michigan holds a very special place in my heart. And I really enjoyed visiting in person about 3 years ago in 2021. Truly had a wonderful time getting to know everyone. And today I'm excited to share even more exciting results that we recently discovered on 3D genome structure behind development and aging. So I've always been fascinated by how cells in our body can develop extremely diverse and different functions, and also age and degenerate differently despite having roughly the same DNA sequence. To give you a few examples of cells that I've studied, the sensory neurons in our nose and eye sense the diverse chemical and physical world around us. Up in the brain, the cerebral cortex is born with very few connections and requires extensive development after birth. On the back of our head, the tiny cerebellum, only 10% of the brain's volume, but harbors 80% of our brain's neurons, and it has its own trajectory of development, aging, and diseases. A common theme behind all these diverse cell types is their very unique 3D genome architecture. So what is genome architecture? We humans have 46 chromosomes, 6 billion base pairs of DNA. All these 2 meters of DNA must be intricately and spatially organized in a cell nucleus of only a few microns in size. And this folding can strategically position genes and their regulatory elements in 3D to control gene expression and regulate our body's immense functions. And this biochemical pathway has been implicated in many disorders, developmental or degenerative. Despite this clinical importance, the 3D structure of the human genome has remained a mystery ever since the 1880s because it's too tiny and too complex for traditional microscope to image. So today I will start by telling you about how we solved the first 3D structure of the human genome by developing new DNA sequencing-based technologies. and how we apply this technology to understand olfactory development and vision development. I will then talk about the brain understanding the dynamics of the genome throughout postnatal development in mouse. And finally, I will talk about our more recent work extending this both across brain regions, across time from development to aging, and also across species between human and mouse to understand the unique mode of development and aging in the cerebellum. So throughout my research, I aim to understand the fundamental generalizable principles of spatial organization of the genome to regulate gene expression, with the long-term goal of precisely rewiring the DNA in 3D to revert disease progression and slow aging. So let's get started with the sensory systems. I will introduce the two types of sensory neurons that I worked on, starting with the nose. So if you stick your finger into your nose, these are the neurons that you will touch. So each olfactory sensory neurons contains hundreds of genes encoding olfactory receptors in their genome, but only chooses one of these genes for gene expression while strictly silencing the others. And these gene expression patterns, we have shown it to be very dynamic during their development and differentiation. And this is a unique system of adult neurogenesis that turn over every few months. At the same time of these transcriptional changes, it has been observed that the shape of DNA also changes dramatically. In fact, starting from 2006, a structural model emerged where hundreds of genes and their regulatory elements from many chromosomes all come together to jointly support this one neuron, one receptor gene expression. But this notion has remained extremely controversial, partly because this structure has not been solved in an actual cell beyond the schematics. The other sensory system senses light. So for many animals, light must pass through the entirety of the genome before it can be detected by the sensory apparatus. And if we slice open the genome, the nucleus of these cells in normal animals like monkeys or us, then the more condensed heterochromatin shown in red here mostly resides on the surface like a normal cell. But in nocturnal animals, like mice and rats, this condensed DNA goes to the inside, presumably forms a tiny lens using DNA to concentrate light at night. This again is very controversial since initial proposal in 2009, again partly because the actual 3D structures of this genome in these cells have not been solved. So what is so difficult about solving the structure of the genome? For two reasons. The one, unlike, for example, protein structures like X-ray crystallography, many identical molecules can be averaged to produce one consensus structure. But genome structure is completely different between cells, even between two cells of the same type. The relative positioning and shapes of all 46 chromosomes are completely different, necessitating a single-cell approach. And that's fine. We must do single cells, and that's what we are very good at with single-cell sequencing. But even within a cell, the 23 chromosomes we inherited from the mother and the 23 chromosomes from the father differ by less than 0.1% in DNA sequence, making them essentially indistinguishable for previous methods, sequencing or imaging. So that is why we developed a new method called diploid chromosomal conformation capture, or DIP-C, to solve these two challenges. So DIP-C is based on the chemistry of 3C and high C, originally invented at Harvard. So the idea is to cut our genome into small pieces while chemically holding them in place in 3D. These small pieces are then ligated to each other based on 3D proximity, forming many of these artificial linkages between two regions that are far away along the linear sequence, but are nearby in 3D, called chromosome contacts. These contacts are essentially the same as distance constraint in like AMR structural determination, but of course on a much more massive scale. In fact, we can apply the same kind of algorithm to convert these proximity measures into the X, Y, Z coordinates of every gene in our genome. So in other words, vDip-C is a biochemical microscope that converts 3D structural information into linear DNA sequence information, which we can then read out with high precision single-cell sequencing. And the cool thing about this kind of microscope without lens is that we're not bound by the physical limits of light anymore. In contrast, everything is tunable by biochemistry. The more context we can harvest from a single cell, the higher the spatial resolution we can achieve. And we have done multiple improvements to really boost the spatial resolution. For example, in traditional Hi-C, biotin molecules is introduced at the ligation junction. But as you can see from this image, the size of the biotin actually is much, much bigger than the DNA it tags. And this would hinder all downstream chemistry. And we realize we can just get rid of this step and therefore really enhance our ability to perform all this proximity ligation. And then we have all these proximity ligation junctions. We also have to sequence them through DNA amplification of the whole genome. At the time, the commercial solution called the Nextera by Illumina utilized TN5 transposase to accurately amplify minute amount of DNA, but half of the DNA will be lost because they only use 2 types of primers. In contrast, we increase this number to 20 primers, leading to only 5% loss, essentially boosting the DNA sequencing sensitivity twofold. With all these great detected contacts, we then have higher computational burden to model the genome at higher scale. To a back-of-envelope calculation, if we model the genome with twice the spatial resolution, it would need almost 4 times the computational resources. And working with my collaborator, Heng Li, now assistant professor at Harvard Medical School, we improved the 3D modeling software, reducing the runtime from multiple weeks to within a day. So together, all these improvements leads to, on average, 1 million chromosome contacts per cell. That is much higher than any previous methods. And here on the left is what the 1 million contacts look like from a single human cell, an immortalized B cell. So every red dot represents 1 gene on 1 chromosome is touching another gene on another chromosome. But this 23 by 23 matrix is not a full picture because we humans have 2 copies of each chromosome, also called the 2 haplotypes. Because the 2 haplotypes are differing so little, only 10% of the read actually carry haplotype information. That means only 1% of the read would carry haplotype information on both sides of the 3D context. And that has always been a rate-limiting step that previously thought to hamper the ability to solve such complex 3D structures. And we developed a new computational algorithm to accurately infer the period of origin of all the context based on the internal clustering structure of the data. Because we see all these contexts are highly segregated on this map, we can aggregate haplotype information from many nearby contexts to jointly and accurately infer which chromosome copy the context come from, turning this 23 by 23 matrix into the full 46 by 46 matrix. And then what's left to do is to get the 3D structure. And conceptually, this is relatively simple. For example, if we consider a small toy chromosome, and if we see the contact map to have an X shape, then we know for sure that its 3D structure is this head-to-tail helping U-shaped structure. Another example, if we see diagonal stripes, that can tell us immediately the structure would be something periodic, like a spring. And note that although simple conceptually, this only works for single cells, because if we add up these two contact maps together, then there won't exist one 3D model that satisfies all these distance constraints. So that really highlights the importance of measuring spatial information from individual cells. And applying this computational algorithm onto a real cell, here is what we get. So on the right-hand side is the 3D structure calculated, not an illustration. It's from the actual data shown on the left. And I'm showing you the 46 chromosomes by different color and pulling them apart so you can see the inside. So this data achieved high spatial resolution of 20,000 base pairs or around 100 nanometers, breaking the optical diffraction limit of light, because we are not actually using light. This is actually just a simple benchtop biochemistry. And we, by undergrad, can do 1,000 of these every day. And in each cell, we can pinpoint the location of nearly 500,000 particles all at once. And this allowed us to recapitulate many of the classic textbook 3D genome conformation. For example, during cell division, in the end, the two sets of chromosomes are put to two ends of the dotted cells in a V shape in human or I shape in mouse. And here we are capturing a dividing human cells in cell culture. I'm showing you one V-shaped chromosome, but almost all the chromosomes look like that. In non-dividing cells, for example, in our blood, another interesting chromosomal conformation is multiple lobes in the peripheral blood. That really helps the cell to squeeze through the vasculature to protect our body. And this is exactly what we saw in cells isolated from a human donor. In mouse, they also have these multiple lobes, but some cells actually have a ring topology. And that's exactly what we saw in the mouse blood. In addition to this very gross scale conformational differences, we can also look at individual chromosomes. For example, if you slice open a structure in the middle and find chromosome 19 and 18, these two chromosomes are very similar in their physical length. But because one of them is very rich with genes and CpG dinucleotides, we confirm the well-known notion that this chromosome, chromosome 19, is almost always in the center, whereas chromosome 18 has very few genes, so it's almost always on the outside. Very consistent with classic DNA in situ experiments. But what's really cool is that we can actually do this for every single location across the genome, achieving subchromosomal localization. For example, the chromosome 1 begins in the center and then goes outside and then center again, offering unparalleled resolution, and also on the whole chromosome scale is highly consistent with whole chromosome painting data more than 2 decades ago. So here, the two copies of chromosome obey the same rules of inside and outside, but the two copies of the same chromosome or genes may not always behave the same. In fact, we are able to visualize two major differences between the two alleles, and one of them is X chromosome inactivation in female cells. So both of X chromosomes are on the surface of the cell because they are very poor in gene density, and as expected. But if you look into the particular shape of these chromosomes, the two alleles differ very different. And only the inactive one, the bar body, creates these super loops across the entirety of the X chromosomes, which was previously reported by Bao Kai-Si. So that really highlights a 3D structural difference between the drastically different gene expression between the two X chromosomes. Another difference between the two copies of the chromosome occurs at a much finer scale. In genomic imprinting, these are genes that are usually very important for embryonic development. There are a lot of local scale differences in gene expression, DNA methylation, and protein binding between the two alleles of the same genes. And for the first time, we can solve their 3D genome structure. And despite the cell-to-cell heterogeneity, once we average a few cells, we can clearly see the differences between the two alleles. And in this case, a CTCF-mediated loop that only exists on the paternal copy of the gene. And I will return to this point later when we have a genome-wide view with much bigger sample size. So in summary about the technology, with vDip-C, we can finally turn all these schematics people have been drawing based on bulk statistical averaged properties of genome structure into actual structures of a cell, of a chromosome compartment, domains, and all the way to loops at the smallest scale. And we have another work that probes even more into these very fine-scale interactions around long genes with our single-cell micro-C method, which is still under revision for nature genetics. So we are now finally ready to apply this technology to the nervous system. And we began with the mouse eye. So shortly after birth, we took out the eye and isolated these structures and sliced them open in the middle so you can see it. Here I'm coloring the genome with its linear sequence property, the density of CpG dinucleotides. And we can see, as expected, the phase separation between the CpG-rich euchromatin in green and the CpG-poor heterochromatin in purple. And we can actually quantify this for every location across the genome. And as expected, we see a negative correlation between gene CpG frequency and gene frequency and the radial inside-outside distribution. So that means the heterochromatin is mostly on the outside, as we expect from a normal cell. But this changes month by month during postnatal development. After 2 months, the whole nucleus essentially becomes this highly regular concentric sphere with the whole genome arranged inside out, primarily based on its linear DNA property, the CpG density. And that presumably positions the denser heterochromatin in the middle of the cell to make it a DNA lens to concentrate light onto the sensory part of the cell to assist night vision. And this is the eye. In the nose, this is a constantly regenerating adult neurogenesis system. So we can look at neurons and their progenitors at any age. So here I'm just showing you one fully mature neurons. At first glance, the purple heterochromatin is still on the outside. So maybe the cell is normal. But the regions that harbors the olfactory receptors shown in red at the bottom is selectively pulled to the inside, away from their regular peripheral localization. And as they are pulled inside, they are also pulled toward each other, forming many of these multi-chromosomal hubs that brings hundreds of olfactory receptor genes and their dozens of enhancer regulatory DNA elements together on the inside of the cell. And this is in sharp contrast to non-neuronal cells isolated from the same tissue shown on the right, where all these genes are both separate and more distributed closer to the surface. And that provides a striking structural basis for their unique one neuron, one receptor gene expression pattern. But these beautiful structures looks nice when we spin it around, But the real question about their functions cannot be answered unless we measure both the structure and the transcriptional profile from the very same cell. And before us, a few months before our work, there has been a few papers doing the same kind of multimodal measurements from the same cell. But the issue is by measuring these two at the same time, the chemistry cannot optimally amplify the two modalities, leading to substantially compromise the sensitivity for both modalities. And this is not enough to reveal the regulatory relationship, especially at the onset of the expression of these genes, when the gene expression is so low. So these other methods cannot detect such low abundance of gene expression. Coming from a physics major, I solved this in a physical way. So instead of trying to amplify two very different things at the same time in the same tube, we can physically separate the 3D genome from the transcriptome via centrifugation. And then we can use highly optimized amplification methods separately for these two modalities, achieving unparalleled sensitivity. And the paper has already been published earlier this year in Nature Methods. So I'm going to only summarize in schematics what we found with this data set. So at the beginning, coming from the adult stem cells into neurons, what we are seeing is these genes behave like normal genes. They have low expression and their enhancers only come from nearby genomic regions. But then they fight. Now they grab enhancers from different chromosomes and fight against each other. And eventually what happens is one of them dominates. And all the other remaining enhancer aggregates are essentially tombstones left behind this RNA-mediated competition, leading to the final form of one receptor expression per cell. And this result is also confirmed by another study by our collaborator using our vDip-C technology. And this is the cover art that my high school student Nick designed for this work, and he's now an undergrad student at Harvard. We have also went beyond mere measurements. We actually also went in to functionally perturb this nice looking structure, both on the DNA sequence determinants of this structure and the proteins that orchestrate them. So I will briefly show you both aspects. So for perturbing the linear DNA sequence, we study a specific subsystems of the nose, that's only 1% of neurons in the nose, called trace-amine-associated receptors. These are G-protein coupled receptors that detect very important odors, such as predator urine, and the one that remains in humans smells rotten fish. So in mouse, many of these genes coming together, they are all arranged in this gigantic 200 kilobase array on one of their chromosomes. And we identify two tiny 2 kilobase regions that are selectively open in their chromatin accessibility in that 1% of neurons. And we use CRISPR genome editing to remove both of these elements, and that leads to completely abolishment of the expression of these receptors without affecting any other genes in the genome. That really showcases the power of long-range gene regulation across hundreds of kilobases. So this is perturbing the linear DNA sequence. We also perturb proteins that are important for genome structure. So for this, we focus on another dimension of olfactory gene regulation, that is the spatial dimension. In case you didn't know, different parts of our nose smell very different things. And as a result, the receptors are actually arranged only in specific spatial regions or zones. They are numbered from 1 to 5 based on their distance from the brain. And all the 3D structures that I've talked about earlier are actually stronger in zone 5, farthest away from the brain. And so is the expression level of 3 transcription factors, NB, NFI, AB, and X. So we actually went in to remove all 3 copies of these gene homologs, and as a result, the zone 5 is now completely abolished, despite no gross morphological changes of the nose anatomically. So the 3D genome structure is greatly attenuated, and then receptors that are originally expressed in this peripheral zone are completely gone. So here, a reduction in the 3D genome structural diversity leads to a reduction in the diversity of gene expression, and finally, a reduction of the chemical diversity the animal can detect through this system. Together, this highlights a strong functional role of genome regulation in 3D in the development of nervous system. So then, having studied the sensory systems, I moved on to the brain, which undergoes a lot of physiological changes from development all the way to aging. And through these processes, the neurons and glia undergo so many different changes, and any changes goes wrong, we would have developmental and degenerative disorders. But inside the nucleus, that's where things get more unknown. Inside the nucleus, 3 intimately connected facets of the genome, its linear sequence or mutations, gene transcription, and genome structure have all been implicated in many brain disorders, but have never been measured before, especially after birth at single-cell resolution. After we are born, the brain grows bigger and bigger, but what is really happening in the genome of individual cells? So let's study all 3 aspects, starting from the linear sequence, which is the most difficult one, because neurons do not divide in the brain. So if they accumulate changes in the linear sequence, accumulate mutations, it only happens in this one cell in this whole brain, and therefore easily overwhelmed by lots of technical artifacts like DNA damage or amplification error during the measurements. So to overcome this, we took advantage of the double-stranded nature of the DNA. So if we can separately amplify the 2 strands of the same DNA, in that way, if in the end we perform the double-stranded complementarity check, we can essentially eliminate nearly all false positives. And we find a way to do this without actually physically separating into different tubes. We can chemically tag the 2 strands of the DNA in the same tube. And here's the result. Without this error correction, previous measurements of mutations in single neurons are all over the place. But with this error correction, we can for the first time measure the accurate mutation number and spectrum of single neurons in the normal undiseased human brain across the lifespan. And we can measure the linear increase in the mutations, about 16 per year, which has important implications for both neuropsychiatric disorders and neurodegeneration. So this is linear sequence. For gene expression and 3D structure, we studied in the mouse initially, because we can densely sample all stages of life, starting from birth. And on the left-hand side, we are doing very traditional single nucleus RNA sequencing. And this plot essentially positions individual cells represented by a dot here and colored by the age of the animal, and cluster them based on their similarities in transcriptional profile. So this is very standard. And in all the major cell lineages of the brain regions we studied, here, the cortex and the hippocampus, we see profound changes in gene expression after birth through development. In neurons, which are the dominant population of these brain regions, we can further delineate different types of neurons' own developmental trajectory, including the hippocampal granule cells, which has adult neurogenesis, which we can recapitulate here. So in neurons, consistent with previous discoveries with bulk sequencing, about 100 genes or so goes down in expression over time, and another 100 gene also goes up. So this is consistent with what people have known before, but for the first time with single cell resolution. So these are transcriptional profiling, which are relatively standard. But from the same kind of tissues, we can also obtain the 3D genome structure. And here I'm showing you 3 representative cells, again colored by chromosomes and rotating, nice looking. But the real question is, can we analyze this kind of high dimensional data systematically, the same way we do for gene expression profile? And the answer is yes, and I'm going to show you one way, is rather than clustering the cells based on their gene expression of this gene high, gene low, gene high, gene low around the chromosomes, I can also ask in the 3D, does the gene reside in the euchromatic A compartment or the heterochromatic B compartment? And this can be quantified with our single cell AB compartment score or SCAP value. Here using the 3D density of CpG dinucleotides as a proxy for euchromatin. So if we feed this kind of SCAP vectors, A or B along the chromosome, rather than gene expression high or low along the chromosome, into the same kind of algorithm that we use for gene expression profiles, then magically these single cell 3D structures, each represented by a dot here, naturally cluster into this 3D genome structure types that are identified through their 3D structure alone, having nothing to do with gene expression. And what's really cool, even more, is that we can match many of these 3D genome structure types into their transcriptional cell types because we find the SCAP value is highly correlated with cell type specific gene expression. So this means we can perform structure typing. Just by looking at the shape of a DNA in a cell, I can tell, is that a neuron or is that a glia? Is that excitatory neuron or inhibitory neuron? So that's truly cool and provides a structural basis for the brain's diverse functional cell types. And this has been extremely useful in the atlasing of the human body, despite coming out almost as a surprise when we first did it in 2018. And in both modalities, a prominent trend is a huge change in both gene expression and 3D structure in the first month of postnatal life. And the natural question is, are changes correlated between these modalities? And the answer is on average, is true. Genes that are reducing expression over time also tend, on average, to move into the more repressive compartment B. But genes that really moves into the more open chromatin compartment A are actually neuronal markers, genes that are upregulated much, much earlier in embryonic development. So that suggests a very interesting temporal mismatch where gene expression changes happens first, and then 3D genome moves around it to further entrench previous cell type commitment. And I will come back to this point later. So these are all compartment changes, A to B, B to A. Another important change, almost even more prominent, is the inward movement of about 5% of the entire mouse genome, moving multiple microns to the inside during postnatal development. And this is completely absent from non-neuronal cells. And interestingly, these are the same regions that moved inside in the nodes that I showed earlier. For example, mouse chromosome 7 has lots of olfactory receptors, and in the nose, and also in the brain, the whole chromosomes can sometimes move entirely to the inside, highlighting an unexpected connection between the two different nervous systems. Finally, we also functionally perturb this structure by rearing the mice in complete darkness for 3 weeks ever since birth and study the visual cortex. And there we see essentially no effect. So the 3D genome progression just goes naturally despite little sensory input from the eye, suggesting a predetermined genetic progression that's independent of early life experience. So together, all this data provides a first look into the genomic underpinning behind our brain's immense plasticity after birth. And from the same data set, we also systematically looked into allele-specific 3D structures behind many imprinting gene loci and provide very cool discoveries. For example, in the PWS8S loci, where we see differences in 3D genome structure between the alleles across the entire chromosomes. So now we have seen development in the mouse brain. What about the rest of the life? In the aging aspect, the neurons do not divide and must work a lifetime after they are born. Yet, previously, despite all the implication of chromatin structure in aging and degeneration, it has never been measured in any organs at all. And one of the limiting factors would be the technology. It has been really difficult to measure many donors of many ages all at once. And all single-cell studies are also plagued by batch effect of processing samples at different time points. So that's why we developed new method, most notably population scale chromosome conformation capture, or POP-C, that we can combine many different individuals in a same pod to measure them all at once, eliminating batch effect and allowing us to profile 3D genome structures at single cell level and at the population scale. And that's how we manage to measure single cell structures across the lifespan of both human and mouse, and in both the cortex that we previously studied, but also the cerebellum, which harbors nearly all the neurons in our brain, but was previously understudied. And here's what the data looks like. Again, each dot represents the 3D structure of a single cell and then clustered based on their 3D structural similarities. And here we can again perform the structural typing, annotating which cell types is corresponding to which 3D structures. And here we are seeing something really, really striking, is that in the most abundant cell types of the cerebellum, called cerebellar granule cells, each of us has like 50 million of them, which certainly tells you how important they are. There we see a profound change in the 3D structure and across a continuous trajectory that unifies development and aging. And the magnitude of this change is many, many times higher than what we have previously seen in other neurons of the brain. Although the other neurons of the brain still follows the same kind of principle, just not as strong as this one. So what is really changing at this massive scale in these cells? So here I'm showing you one representative chromosomes. I'm zooming in also into one locus. Here I'm showing the homologous locus between human and mouse. So around birth time, these neurons look like a typical neuron in the sense their 3D genome structure is not prominent. Genes essentially only touches genes if they are nearby in the linear sequence. And that has been a characteristic structure of both embryonic cells and neurons. But as time goes by, both development and aging, this structure changes completely. Now we are seeing the emergence of extremely long-range contacts pulling together in 3D genes that are 1 million, 10 million, or 100 million base pairs away, forming this strong checkerboard pattern of the diagonal, suggesting very strong phase separation between the newly formed chromatin compartments. And we can clearly see that this pattern is highly conserved between human and mice, despite the fact that mouse only lives for 2 years and we live for 100 years. That's actually a great property for the mice, suggesting they are, in fact, a very good model of these 3D genome changes in age. This is almost a clock-like phenomenon. By looking at the shape of the chromosome, I can tell how old the person is. Is the person in the 20s or in the 60s? Almost like a 3D genome structure clock. This does not only happen within a chromosome, this can actually happen between chromosomes too. Here I'm showing you the human 3D structure, but we have found it to be conserved in many other organisms, that selective pairs of chromosomes are also put together to form these very specific inter-chromosomal interactions. And that is really exciting for us because people have thought for a long time that all these interchromosomal specific interactions is just a curiosity that only happens in the nose. But in fact, this is quite generalizable. So what does this restructuring do? And we can only begin to hypothesize based on its relationship to gene expression. And here's an example of what we typically see. So here, GABA6 is a very important GABA receptor that are only expressed in the mature form of the neurons, which occurs around birth time in humans. What happens is, although the gene is upregulated around birth time and stays relatively constant over time, the 3D structure continues to change over decades after the initial upregulation. So that essentially means that the strong cell type specific gene expression continues to slowly etch the 3D genome structure around it over 100 years. So that's what we call a transcriptional etching, and we are just in the beginning to understand the meaning of this and how we can manipulate it. So in summary of my talk, we developed a DNA sequencing-based technology called vDip-C that solves the first 3D structure of the human genome in a single cell. And we initially applied it to study cell lines and blood cells in humans. We then applied it to the sensory systems, discovering highly specialized 3D genome structures in the nose and eye behind their highly specialized sensory functions. We then moved on to the brain, and there we see lifelong genomic dynamics in the accumulation of somatic mutations along the linear sequence, changes in gene expression, and lifelong remodeling of the genome structure that are conserved between human and mice. To put this into a bigger perspective, ever since the 1880s, scientists have been looking at our genome under a microscope, wondering about their 3D structure. 2003, the Human Genome Project mostly finishes the linear sequence of the genome. But now with vDip-C, we can finally see its 3D genome structure. And with only a few examples that we have studied, we have already observed such great diversity and specialization of genome structure across development aging. And we are barely scratching the surface here to understand 3D genome regulation. And I would like to thank my lab, founded in late 2022, and my mentors, collaborators, my founding sources, and thank you so much for the invitation and for listening.
Thank you so much, Longzhi, for the very wonderful and exciting talk. Now we have 10 minutes for Q&A. Please feel free to unmute yourself to ask a question or simply type your question in the chat window. I can probably get started with the first question. So I am very curious about what accounts for the structural stability of the genome in the cell. For example, in the one cell, one receptor case, once the enhancers bind to the gene and decided to amplify, really to activate that specific gene, what stops them from really moving around to activate other genes? Yeah.
That's right. Yeah, so that has always been extremely mysterious. And in the past, we have find a few proteins that stabilize this conformation, but mostly through just the very crude try one gene at a time approach to delete it and see what happens. And so us and our collaborators have find like transcription factors, protein-protein interactions, or chromatin modulators that can actually stabilize the structure in the sense that if you delete this, then it forms a very strange state. Either can never settle on one, or they have multiple. But it has indeed been challenging to study because unfortunately, this olfaction is extremely important for mice. This is in sharp contrast to humans, where about 5% of us just don't smell anything. I think olfaction is really brought into spotlight by the whole COVID thing, where our collaborator actually shows that the 3D genome attenuation is actually behind partly the smell loss. In mouse, perturbing it makes them bad, essentially, because they are born eyes closed, and without it, they cannot find milk. It's called competitive suckling defect. So we have indeed lots of mutants that just don't survive at all. So that has created lots of difficulties. And we are really hoping to build more high throughput method that we can screen for unstable conformation in the end as some sort of phenotype that would be really amazing. And we're trying also a lot, like how do we deliver a virus into those things? And they also have weird properties that's not as manipulatable as other systems, but we are still continuing some of this line of inquiry, mostly in the form of collaboration.
Got it. Wonderful. Thank you.
Okay, yeah, I have one question. Thank you very much for the very fascinating talk. Yeah, it's really
Thank you.
Yeah. So, okay, so you show the 3D structure of each chromosome. Then how much resolution do you think you can expect? In other words, so let's say you see the curvature. Then how much precisely can you identify that sequence in the chromosome?
Yeah, that's something we are hoping to improve the spatial resolution. And right now, most of our published structures are about 20 kilobases in sequence. And then for the single cell micro-C one, we do have higher resolution. So some of the structure can achieve about 10K or 5 KB. And at the same time, we do have footprinting if a protein binds to it. So in the single cell micro-C data, if there's a protein like a CTCF protein, then we can actually see there's a footprint in the 3D structure. But we are indeed actually missing this footprint to 5 KB resolution that I think is actually a very important gap in our knowledge about chromatin in general, between like fine scale things about chromosome remodeling, chromosome accessibility to all what we measure a higher order chromosome conformation on the kilobase or tens of kilobase scale. So we hope one day we can bridge this resolution gap, since there is not really any true dichotomy between like single binding to higher order measurements, other than our inability to build tools to span both. So if we can take this idea of a biochemical microscope to this kind of resolution scheme, we can hopefully one day build this kind of structures of every single nucleosome in a cell, but we know they are sequenced, unlike in EM graphs. So that can really provide a holistic view, hopefully down a few years in the future.
Okay, sounds good. Okay, I have one more question. So yeah, another the fascinating part is that the inter-chromosomal interactions. Yeah, that's quite amazing. Yeah. So then, do you see the kind of the same region interact each other in, or that is pretty much context dependent?
Yeah, so that's the thing we're still understanding. So in the olfactory system, what's happening is, at least in the chromatin accessibility side, there are lots of these 1 KB enhancers that are very prominent in the structure. So in fact, if we look at carefully close the contacts, there are always a very strong entanglement of the actual enhancer themselves. And then the nearby genes, which are much longer, then they almost form like a surrounding sphere next to it. And our collaborator and us, we did have disrupt some of these very focal interactions. So in the TAR system, which I showed, it actually works pretty well. That it's actually the very small contacts that anchors everything. If you get rid of them, the whole thing falls apart. We haven't really achieved the same thing for the 1,000 receptor genes. Our collaborator has disrupted a few, and it doesn't have as strong an effect. It's more like you cut off this one, then this small part falls off, and then cut another falls off. We haven't reached the criticality yet, but it's also limited by how many genes we can remove by CRISPR at the same time. So I think one day we might get there. It's like almost emergent phenomena that you need how many enhancers altogether to abuse it. And then in the brain, and there's also a concurrent preprint from Azad Boni's lab. So it seems the trend is there are cell type specific genes, and they are highly expressed in the cell types, and the gene and genes seem to form very strong contacts. And in this case, these are actually longer genes than the olfactory receptors. The olfactory receptors are just GPCRs, but these non-neuronal genes are actually pretty long, and they entangle a lot. And then all the heterochromatin next to them then forms really large clouds of interaction. So almost it feels like there's a cloud of heterochromatin that freezes the tiny genes in place. So we are thinking maybe we can also do the same to disrupt the core. But another thing we notice for the brain is that the heterochromatin part is really great. There are a huge amount of them. So another hypothesis we are thinking about is that they also serve the purpose of silencing the heterochromatin more efficiently. And at the same time, consistent with this notion, the cells become smaller, and also they're more heterochromatinized at the same time. So we are thinking maybe over age, they really want to prevent unnecessary gene expression. So that's maybe a way to silence all the transposons in our genome. But that we still need more validation of this hypothesis. But the idea is maybe as we age, the cells are more and more narrowed into what gene they can express, and they don't want to express other things. And one way of rejuvenating the brain maybe is to reverse this process, and then I can unlock genes that the cell normally don't want to express for wound healing purposes, maybe. But these are speculation at the point.
Okay. Thank you. Thank you so much.
Thank you very much.
Okay, I probably can ask one more question. So, I think the ligation process could be very important for the DNA microscope. So what do you think is the limitation of the ligation efficiency? Because I can imagine if the two DNA ends is like wandering around, right, there could be some percentage of chances, like 90% or 80%, they can be finalized into ligated single or double strand DNA. So what do you think are the limitation of the efficiency? Thank you.
Yeah, so that's a great technical question. So indeed, I think there are lots of rate limiting steps, both in digestion and ligation. And I think to achieve more, we really need to tune the landscape of all these reactions. So right now, both digestion and ligation are actually limiting in the sense that the digestion also needs to happen frequent enough for it to create free ends. And that has been quite limiting because we are only cutting about every 1 kb to 2 kb. That's much, much sparser than the actual density of the restriction site. So that's one thing we are seeing. That's why in the micro-C site, so single cell micro-C, we are using mononucleus to cut, which are much more efficient in cutting. But at that point, we're running into the opposite issue that the mononucleus actually has strong DNA degradation activity, unlike restriction restriction enzyme, it never degrades the DNA, but the mononuclease actually chews up. So that seems to have an issue. Maybe it chews up too much, then it doesn't stretch enough to ligate. It also creates lots of single-stranded overhang, which cannot be ligated with most ligases. So that has been quite challenging. So in general, there is also a problem of how precise the DNA we want to measure. If interaction is very fine scale, if we disrupt it too much, then we can ligate, but it essentially changes the structure, which I sometimes call the Heisenberg uncertainty principle. To measure it, you have to disrupt it. So we really hope to bring in a lot of like hydrogel chemistry, local amplification to make it work much better, like what Lab is working on. So this kind of thing can really help to both fix the things in place and create more junctions, and maybe put filler sequence in to bridge potential distances, almost like a ruler-like fashion. So I think there are lots of chemical improvement that the field can focus on, while we also apply the tools to understand neurobiology.
Great, thank you. I'd like to thank Dr. Tan again for the wonderful talk, and I will thank everyone for attending, and I hope everyone have a wonderful day. Bye.
Thank you so much. Take care.
Bye.